You squeeze sideways through a crack barely wider than your chest. Your shoulders scrape stone. Your light catches a human skull. Then another, then another. Deep inside a South African cave, 20 ancient individuals left behind teeth that still carry molecular clues. Every tested one points the same way: female. Not one produced a confident male signal. That does not mean every Homo Naledi was female. It means something far stranger happened inside this cave. And the answer may change how you picture an ancient mind. The story begins in 2013. Inside the Rising Star cave system near Johannesburg, explorers entered a chamber called Naledi. Getting there was brutal. The route twisted through narrow passages. One squeeze was only about 18 centimeters wide in places. That is narrower than many human chests. Beyond it sat an extraordinary fossil collection. In 2015, Lee Berger and a large international team announced a new species. They named it Homo Naledi. Homo means it belonged to the human genus. Naledi means star Suthu. The name came from Rising Star itself. The first collection included more than 500 fossil pieces. They represented at least 15 individuals: adults, teenagers, children, infants, a whole range of ages. But their bodies looked like a puzzle built from different eras. Their feet were surprisingly human-like. Their legs suited upright walking. Their hands could grip and manipulate. Yet their shoulders and curved fingers looked more ancient. Their brains were tiny, roughly 1/3 the size of yours. That combination made no sense under the old ladder of evolution. Human evolution was never a ladder. It was a crowded bush. Different branches kept different traits. Homo Naledi proved it dramatically. Then came another shock. In 2017, Paul Dirks and his colleagues dated the fossils. They placed Homo Naledi between about 335,000 and 236,000 years ago. That was astonishingly recent. These small-brained relatives lived while early Homo sapiens already existed in Africa. Same continent. Overlapping time. Very different bodies. But the deeper mystery was not their anatomy. It was their location. The fossils were not scattered near an open cave entrance. They were deep underground. No obvious river had washed them there. No large predator den explained the pattern. The chamber contained almost no remains from other large animals. So how did so many bodies reach that darkness? One possibility was deliberate disposal. Perhaps Homo Naledi carried their dead inside. That idea sounded radical. A creature with a brain that small was not supposed to manage complex treatment of the dead. But supposed to is where science often gets trapped. In 2025, Berger and colleagues published a revised study in eLife. They described concentrations of bones inside sediment-filled features. Some body parts remained articulated. Articulated means the bones still sat in their natural anatomical order. That usually suggests soft tissue connected them when sediment covered them. The researchers argued that the bodies had placed into excavated pits, then covered before decomposition. That would make them burials, not accidents, not bodies slowly buried by cave dust. Burials. The claim remains debated. One eLife reviewer considered the evidence convincing. Another said the case was still incomplete. That disagreement matters. Science does not become weaker when experts argue. It becomes visible. You can see exactly where evidence ends and where interpretation begins. Then in 2026, the cave produced a new kind of evidence. Not another skull, not another pit. Proteins. Balesa Madupi and an international team examined dental enamel from 20 Homo Naledi individuals. Dental enamel is the hard outer shell of a tooth. It protects tiny protein fragments for immense spans of time. DNA often breaks down. Enamel proteins can survive longer. The team focused on amelogenin. Amelogenin helps form tooth enamel. Two versions can matter here. AMEL-X comes from the X chromosome. AMEL-E comes from the Y chromosome. Find AMEL-E and you have strong evidence of a biological male. The researchers sampled 23 teeth. Those teeth represented at least 20 individuals. 19 were classified as female, with more than 95% probability. The 20th was probably female too, but its result was less certain. Across the entire set, the team found no confident male marker, no amely, not once. That result was so unexpected because the sample spanned the cave system. It included different ages, different chambers, different body sizes. Even Neo was included. Neo is the remarkably complete skeleton from the Naledi chamber. Researchers had often pictured Neo as male. The skeleton looked large and powerful. But the proteins pointed to female. That single result exposed a quiet assumption. Large meant male, small meant female. It felt reasonable, but it had never been proven. The fossils had very little size variation. Researchers once used that similarity to discuss the species as a whole. Now, the molecular evidence suggests they may have been measuring mostly one sex. That changes everything. The average body size may be wrong. The expected male body may be unknown. Even reconstructions of Homo Naledi could carry modern bias. A broad chest does not automatically mean male. A large skull does not automatically mean male. The cave had preserved ancient bodies. It also preserved modern assumptions. So why were the tested individuals female? There are several possibilities. The first is simple sampling. Maybe males are present. Their teeth may not have been tested. Some known individuals are represented by bones without suitable teeth. Future analysis could find amely. 20 individuals sound like a lot. For ancient molecular research, it is enormous. For an entire species, it is tiny. So the phrase matters. All tested individuals were female. Not all Homo Naledi ever found, not the entire species. The second possibility is biological. Perhaps amely was missing or altered in Homo Naledi males. Modern humans can rarely lose parts of the Y-linked amelogenin region. If Homo Naledi males lacked the marker, the method could misclassify them. But the researchers examined more than one line of protein evidence, and no tested individual gave a confident male pattern. The biological loophole exists. It just does not erase the pattern. The third possibility is the most provocative: selection. Someone chose which bodies entered these chambers. Not randomly, not equally, by sex. Imagine the scene. The sun disappears outside rising star. A small group gathers near the cave entrance. One body lies wrapped, carried, or supported. The group enters darkness. They move slowly. Hands press against cold walls. Feet search for ledges. A flame flickers. Smoke collects above them. The passage tightens. One person exhales and turns sideways. The body must pass too. Every meter becomes a decision. They keep going. Eventually, they reach the chamber. Perhaps sediment has already been disturbed. Perhaps a shallow pit waits. Perhaps this is not the first journey. They lower the dead. They cover her. Then they return toward daylight. Nothing in that scene is proven. The proteins cannot show grief. The bones cannot reveal a ceremony. The cave cannot tell you what anyone believed. But the physical problem is real. Bodies reached a difficult underground space, and the tested teeth belong only to females. If that pattern reflects behavior, then Homo Naledi may have practiced sex-specific mortuary treatment. Mortuary means what a group does with its dead. That could include burial, placement, removal, protection, or something with no modern equivalent. This is the twist. The all-female pattern may not be about females at all. It may be our first glimpse of a social rule. A rule made by minds unlike yours. Researchers often search ancient bones for anatomy. How tall was this species? How heavy? How did it walk? But a biased group of bodies can preserve behavior. Who was selected? Who was excluded? Who belonged in a particular place? The missing males may be the most important fossils in the cave, because absence can be evidence. Not proof. Evidence. Perhaps males were placed elsewhere. Perhaps female bodies held a special role. Perhaps kin groups used different numbers. Perhaps the cave represented age, lineage, status, or danger instead of sex. The protein result narrows the mystery. It does not solve it. Before the protein study, researchers had already noticed something odd. The adults looked unusually similar. In many primate species, males and females differ in average size. Scientists call that sexual dimorphism, a technical phrase for bodies shaped differently by sex. Homo Naledi showed surprisingly little of it. That could mean males and females looked alike, or it could mean researchers were comparing females with females. The new evidence makes the second option impossible to ignore. It also changes how fossils are displayed. A museum reconstruction looks authoritative: hair, skin, muscle, a face staring back at you. But every layer contains choices. Artists choose body mass. Researchers estimate sex. Writers choose pronouns. Those decisions harden into public memory. Neo became a powerful example. One of the largest skeletons looked male to modern eyes. The enamel disagreed. That is not a minor correction. It is a warning. Fossils do not arrive with labels. The labels come from you. The cave also forces another question. Could 20 females accumulate there by chance? Imagine drawing 20 births from an even population. The chance of selecting the same sex every time is tiny. But this was not a clean random sample. Preservation was uneven. Excavation was incomplete. Not every body left a testable tooth. Not every tooth was sampled. That means a simple coin flip calculation would mislead you. The sample was shaped by death, by cave movement, by preservation, by excavation, and by laboratory access. Still, the pattern demands an explanation. Randomness is possible. Indifference is not. Future work can test more teeth. It can search for rare Y marker loss. It can compare chambers separately. It can revisit body size estimates. It can ask whether age and kinship follow the same pattern. One male marker could transform the story. A larger female sample could strengthen it. Either result would matter. That is how a real mystery grows. Not by protecting one dramatic answer, by making every answer survive the next test. And that uncertainty is exactly why the discovery matters. For years, Homo Naledi was treated as a contradiction. A small brain, a human-like foot, ancient shoulders, recent dates, possible burials. Now add a female-only tested sample. Each clue resists the same lazy equation. Bigger brain equals richer behavior. Modern body equals modern mind. Large skeleton equals male. Simple anatomy equals simple society. Those equations feel neat. Nature was not neat. A brain roughly 1/3 your size could still navigate a cave. It could recognize a dead companion. It could follow a shared practice. It could possibly distinguish where different people belonged after death. That does not make Homo Naledi human in the modern sense. It does something more useful. It makes the word human harder to define. Your own life is full of invisible sorting rules. Some people enter certain rooms. Others do not. Some names are remembered. Others vanish. Some bodies receive rituals. Others receive silence. You inherited those behaviors from cultures built long before you. The rising star cave may preserve another culture's rule. A rule separated from you by nearly 300,000 years. You cannot read it. You can only see its outline. 20 tested individuals. 19 confidently female. 1 probably female. 0 confident males. A narrow passage. A chamber of bones. And a choice someone may have made in the dark. You entered the cave looking for bodies. What you found was a rule.